name is Bruno Musch. I'm a neurologist. I am at the 5th Congress on Sleep Medicine here in Valencia, Spain. And we have the pleasure to have with us Dr. Amanda Freeman, who is instructor at Emory Medical School in the United States. And she is very involved in some fundamental research about animal models. And she will tell us something that you will find absolutely fascinating. So, Dr. Freeman, can you just summarize what is your research field and the data that you're presenting here in Valencia?
Sure, thank you. Genome-wide association studies have identified 6 different loci that have been very important or at-risk genes for RLS and PLMS. However, the molecular role of these genes and proteins in causing RLS is still unknown. What's going on in the cell? Why do these genes, these variants, actually lead to RLS? So we've turned to the Drosophila model because of its genetic toolbox and ease in altering gene expression to look at what may be going on. And first, we actually focused on BTBD9. This gene, when it was identified, simply had a name. No molecular role was known in flies, in humans. It hadn't been studied anywhere, but it accounted for 50% of the population attributable risk of RLS. So when we knock that out of flies, we find that they have very fragmented sleep, but there's no difference in how much they sleep over a 24-hour period. However, at night, their sleep bouts are very short in length, and they have a lot of sleep bouts. And once they fall asleep at night, they continually wake up and have a lot of wake after sleep onset at night. So very fragmented, as you would see in our RLS patient. In addition, they have hyperlocomotion. And if I put them in a very confined space, as if you would ask an RLS patient to sit still and not move, which tends to aggravate their symptoms, these flies will move more, 2 to 4 times as much as a normal fly. They're not moving faster, they're not hyperactive in that way, but they just don't stop to rest. They just continually move while normal flies would sit still. We know that we can treat RLS patients with dopaminergic drugs. So we were curious as to how BTBD9 would actually interact with dopamine. And one advantage that the fly gives us is that we can not only control gene expression across the entire animal very easily, but we can control it in very specific cell types. So if we knock down BTBD9 expression in all neurons, we can get the sleep fragmentation, the same sleep phenotype. But if we restrict that knockdown to just the dopaminergic neurons, we are also able to get that. So that suggests that BTBD9 in dopaminergic neurons is key in regulating sleep consolidation. It's very necessary for that. If we treat RLS patients with dopaminergic agents, we asked, can we actually treat the flies? So we put pramipexole in the fly food. And indeed, on a day by day, as they increase their dose, because they're continually feeding on this food, their sleep fragmentation gets better and better. And by 3 days of feeding on this food, their sleep fragmentation has been rescued. They're down to what looks like a normal fly. So we know that it interacts with the dopaminergic system. Studying iron in a fly is sort of difficult. It's a young field. They have a different buffering system, so altering iron in their diet doesn't have the same effects on the central nervous system that it does in other species. So we turn to the cell culture to look at how BTBD9 interacts. And using human embryonic kidney cells, we transfect them to overexpress BTBD9. In those cells, you see an increase in ferritin just under normal baseline conditions if BTBD9 is increased. So therefore, we can see that BTBD9 expression is increasing ferritin expression. If we alter the iron environment of the bath in the cell culture, we can also then alter ferritin expressions in combination with BTBD9. So we've shown that, like RLS patients, the flies without BTBD9 have sleep fragmentation, they have altered locomotion, or hyperlocomotion when they're in confined spaces. It interacts with dopamine and it interacts with iron. So we've replicated this and established that we have a viable model of RLS in a fly. We're now looking at other genes that have been identified through genome-wide association studies to see if we can identify how the different genes may interact to give rise to the different phenotypes. Since RLS is such a heterogeneous disorder, can we tease apart certain patients that may respond to certain drugs, may show augmentation differently, may have different characteristics based upon the underlying genotype. So now we're looking at PTPRD and have shown that with loss or overexpression of PTPRD, we can see the same sleep fragmentation, and now we're looking for other characteristics.
Well, thank you very much. I think that I'm sure you are as fascinated as me to see how much we can do with flies, but particularly how relevant are these animal models in the way we're trying to understand the disease, with also implication the way we are going to treat the disease, particularly in the search of new treatment and new benefit for our patients. Thank you very much.
Thank you.